I really like to thank the organisers. I'm not a vet, but I work at the Royal Veterinary College when I've been working there for 20 years. Now, vets are great, but I have to say, vets are cool. What a great time I had yesterday. So it's a real pleasure. I'd really like to thank the organisers for inviting me to speak to you about the immune system, which has taken leaps and bounds. And I'm just going to give you sort of a bit of a snapshot about what we know about immune aging. And then you're going to hear from the real scientists, Professor Powlett, Dr. Charles and Dr. Dougal. And in part, it's going to be related to what was spoken yesterday in terms of the gut microbiome and nutrition. So please do keep those questions. I realize we've got an hour Q&A. So I do hope you're able to provide those questions. As customary, these are the opinions of myself, or not necessarily that of Nestle. This is my disclosure. One of the things that's really important, and I think we're now starting to very much appreciate, is the importance of our immune system. And what myself and what my fellow speakers really want to get across in this session is the relationship really between sort of immunity and functional immunity and longevity and healthy aging. And there's a real sort of connection with that. Because what we're starting to see, and this was mentioned yesterday, it's something we're now, I think, very appreciative of, is the fact that as we get older, there's a series of chronic diseases that becomes apparent. And I know Professor Powlett Graham will be talking about this whole concept of what we called inflammaging, which basically, as we get older, there's an increased production of pro-inflammatory cytokines. And basically, it's the fact that the body is swimming in this high level of pro-inflammatory cytokine that gives rise to a number of these, chronic diseases. Not only is that seen with age, but also, and this is a slide just taken looking at, just under 20 years ago about flu. And I know, again, Graham and the rest of my colleagues will be talking about this. So what you see with increasing age is the fact there's an increased morbidity and mortality. And what you find with that is an increase in, sort of disease infection. This is with flu. And sadly, this is the data from COVID. And as you can see, and I'm sure we're all aware of the fact that age is a major risk factor, sadly, with COVID. And so the immunological community have decided over the last few years to really sort of focus on this important area. And what we spoke about yesterday, particularly the fact that there's an increasing aging population, and I think I saw a slide that mentioned, coming up to 20% of the population in 2050 or 2060. And so immunologists are starting to look at this. And in fact, there's a couple of journals I really want to point out to you. So there's Trends in Immunology that's been going for a number of years. And it's the very first time they dedicated an entire issue to one particular theme, and that was immunosenescence. And this is basically the kind of working definition. It's the age-associated decline of immune function that predisposes individuals to increases in infection disease, which is associated with morbidity, mortality, increase in cancer, and also less responsive to vaccination. And then a few years later on, clinical experiments in immunology, again, devoted a special issue. So immunologists are taking this really seriously. And what we're starting to see, for example, is a reduction in fighting diseases. It's not completely lost, and it's really important to recognize that. But it's taking time to recover. But again, it's this inflammatory increase in disease. And what's really quite interesting, I spoke to a group of rheumatologists, and perhaps this is similarly in the veterinary profession, that disease-modifying agents that's been now used to treat a number of these sort of chronic diseases are sort of compounds that are anti-cytokine reagents. So realizing the idea of the importance of these inflammatory mediators. And also, sadly, the poor response. So what you see, for example, with flu, And data suggests that somewhere in the region between 35 to 50% of older individuals, have a poor response to the vaccination of flu. So what people have been doing, and what I'm going to talk about, is just some of the sort of changes that's seen in the immune system, that's in blood. And then I kind of want to focus on a bit of a hobby of mine, which is the thymus, the organ that's responsible for making T cells. Because what I really wanted to inform you is that I think the thymus is going to be a key biomarker in aging. And I'm sure some of you are aware of this. And in particular, Dr. Dougal's going to be covering this. So here's some of the important and overview features that we see. A lot of focus on sort of declining immune function has been focused on the T cells. T cells sit in the center of the immune system, viewed as the orchestrator of generating an immune response, helping B cells, helping the innate immune system. So naturally, there's been a focus. And I just wanted to show you that this kind of feature here is the fact that if you compare young T cells to old T cells, a key thing, particularly is functional, is the fact there's a reduction in receptor diversity. And a lot of folks have been using that as a hallmark. So a decline in receptor diversity is such that it may give individuals an increased susceptibility to disease. There's proliferation defect, and that's associated with telomere shortening. And again, one of the functions that T cells do is to support and help the immune system, and an example of that is B cell collaboration, which again is slightly declined. B cells also undergo changes. What you see, for example, is the developmental changes in B cells, but also class switching. So class switching is not effective from an IgM, from a primary to a secondary immune response. Even the generation of a secondary immune response, there's suggestions to suggest that there's a reduction in high affinity antibodies. And high affinity antibodies is really key, particularly in terms of, preventing disease and vaccination. There's also changes in innate immunity. And what people have identified is that in hematopoietic stem cells, they noticed that with increasing age, there's an increase in various senescent markers like P16. But what's quite interesting, that hematopoietic stem cells lose that potential to develop into lymphocytes and are actually skewed towards more of a myeloid differentiation phenotype. And what's quite interesting, I was looking into this recently. I don't know the situation with other countries, but in the UK, once you're over 50, you aren't necessarily considered to be a donor for bone marrow. You can still give blood up to 70 or whatever, but not with bone marrow. And that's quite interesting to suggest that the differentiation capacity of aged stem cells is somewhat reduced. A lot of work has been done looking at innate immunity. And we've looked at, for example, natural killer cells. And one of the things we've observed with natural killer cells is there's a change in phenotype and function. So overall, there's many changes with the innate as well as the adaptive immune system. But what I want to focus on for a couple of minutes is this really important area of thymus biology. So the thymus has been known for many years to be the university of what we call of T cells. So stem cells will migrate into the thymus where they receive all the signals to become functional T cells. If you don't have the thymus, you don't make T cells. And so for many years, people said, that's it, we know how thymus and we know how T cells are developed. And several years later, people are starting to look again at this organ, which is situated above the heart. Because what people have noticed, and this has been known for a long time, even in Victorian times, they noted that when you looked and did autopsies in older individuals, they noticed that's this little shriveled organ. They didn't really think much of it at the time. But now more and more people have identified that with increasing age, there's this situation of age-associated thymic involution. And moreover, it's really one of the hallmarks of an aging immune system. And what you see, and this is some studies we've been doing, is that we've got a mouse colony. And what you can see is that there's a decline in the number of total thymocytes. So the mice were used for different studies. We were fortunate that no one else was working on the thymus. So we were able to collect the thymus and literally just count the cells. And as you can see, there's an age-associated decline in thymic numbers. And what's quite interesting, I want to come back to this, is that the decline is actually not steady state. There's actually a rapid decline early in life, and then there's a slow decline subsequently, and that's something we're going to come back to. People have been looking at age-associated thymic involution in a number of different species. In humans, you see the same, where it's a change in the fact that there's increasing adipoticity. We've been fortunate to look in dogs. As I said, I work at a vet school, so I was managed to meet some wonderful vets like Holger. And what we were able to show in, this is in Labrador dogs, is there's an assay that you can do in humans called the TREC assay. It's almost like a default in T-cell development. So what happens when you are making T-cells, there's this basically just a bit of DNA, this circular DNA, a bit like plasmid. That's a residue byproduct of when T-cells are being made. Moreover, you can actually use that as a biomarker to measure thymic output. And what you can see is that in Labrador dogs is the fact that there's again an age-associated decline in what we call TRECs, which basically means there's an age-associated decline in producing naive T-cells. So again, like other species, age-associated thymic involution exists, even to the point where zebrafish, which is the most primitive animal that has a thymus, you can see here from histology that the thymus involutes. So this process of age-associated thymic involution is evolutionary conserved, and that's something that's really fascinated me for an incredibly long time, and I'm really fascinated to understand why that is and what does it mean. And here's what it kind of means in the periphery, and I'll explain this. So what you find, and this study's been done in mice, but a lot of work has been done in other species, is in young animals you find that this population of naive T cells literally decline with age. It doesn't mean, and it's really important, that T cell numbers decline with age. In fact, T cell numbers remain consistent. But it's the fact that there's a lack of naive T cells that T cells change their composition. We also see the same in dogs. And the real problem, and I'm really grateful for the folks from the Dog Project, and one of the things which is a challenge, particularly for looking at this in companion animals, is there's not appropriate antibodies to try and identify different T cell population. So again, we've been using PCR methods, and what we've shown, similar in humans and similar in mice, is that with increasing age, as I said before, there's TREC assays, but also there's a reduction in T cell repertoire. This is by using what we call spectrotyping. This is also seen when we've been looking at peaks. These are just methods to identify receptor diversity. So again, it's following a similar pattern, that there's thymic involution and there's a decline in TCR diversity. And diversity is the key in mounting an effective immune response. It's seen as a risk factor. I know, as I said, Professor Pawlak and others will be exploring this. So for me, I've been trying to, and for others, we've been trying to convince the immunological community that the thymus is itself is a real trigger of immune senescence. And I'm hoping at the end you might think this. So here's what happens, what we're now thinking. So if you're a young individual, your thymus is churning out T cells. So here's what the peripheral pool T cells look like. There's a plethora of diversity. Diversity is key. So you have some cells that have left the thymus, recent thymic immigrants, you've got some naive T cells, and you've got cells that are memory and activated. What happens as you get older, the thymus shrinks, and the impact of that is the fact that the peripheral T cell pool doesn't change. There's a process known as homeostatic proliferation, where in fact memory T cells will take up that space that's not going to be occupied by naive T cells. And so one of the drives for that, and maybe Graham might be talking about that, is CMV. But equally, what also happens, and I hope everyone's colorblind, is to see there's a reduction in diversity. But also, because of this homeostatic proliferation, these memory cells want to occupy that space, you also have an exhaustion of cells. So cells often have what we call, telomere shortening. So the thymus is intricately linked with immune senescence. And so over the years, people have been trying to identify, what are the mechanisms? So is it factors inside a cell, such as telomere shortening, such as the changing in the phenotype, from having more memory cells, having a reduce in TCR diversity. But there's also some really interesting studies to suggest there's also environmental factors. The cells are sitting in a higher level of pro-inflammatory cytokines. But even the microenvironment which cells sit in order to differentiate and actually proliferate is also altering. And I want to show you some really nice sort of pictures associated with that. So here's what we're thinking is happening in the thymus in terms of thymic involution. As I said, stem cells from the microenvironment, from the bone marrow, enter into T cells, into the thymus, where they receive all the signals to become functional T cells. If you don't have a thymus like DiGeorge syndrome or SCID, you don't make any T cells. And again, similar situation seen in fox and the nude mouse. And that's also seen basically in the aging thymus. What we're seeing is the fact that in the aging thymus, the microenvironment is pretty much defective, which therefore leads to an alteration of thymopoiesis. And so we've also been looking at the secondary lymphoid organ. I just want to show you some pictures because I think these pictures are absolutely fantastic in looking at the microenvironment, because cells don't make immune responses in blood. They make immune responses, as I'm sure you'll know, in lymph nodes. And we really caught our attention to this because there are some mutations in mice, the ALA mutations, etc., that don't affect hematopoiesis, but actually affects the structural organization of lymph node. And that got us really interested to look at this. And so here's one of the things you'll see. In order for proactive T cell and B cell to become activated, there needs to be an engagement together of cells. T and B cell collaboration is really important. And what you find in lymph nodes is they're well organized and well structured. You see there's T cells and there's B cells, as illustrated here. What happens as you get older, you see this complete dysregulation. And with that dysregulation, as I'll show you later on, there's also an alteration of immune response. We did quite a lot of work in order to get this. So we did a lot of sections, and we did sort of image analysis. So it required a lot of validation, and my PhD student, did an incredible job. We also see alteration in the stroma, the environment of which these cells resides. We also see alteration in dendritic cells. Dendritic cells are really important because they are kind of the initiator of an immune response. And the characterizations of these cells are completely sort of dysregulated in older spleen. And these marginal zone macrophages that sit around the white pulp, which are surveying sort of the antigenic experience of mice, is also altered. And you see an increase in fibrosis in aging spleen. And you also see the same with lymph nodes. So it's not just the fact that you see this in spleen, and what you see, this is what a young spleen looks like. And for those of you who are familiar, it's the idea you've got T and again B cell zones. And what you see with age is the fact that this is completely altered. And this is really interesting. So this is what it looks like in young, double staining. B cells are in green, T cells are in red. And then what you see in aged, they're completely all over the place. People have done further studies where they've done adoptive transfer experiments. And what they've shown is that if you put young T cells into an aged spleen or in an aged lymph node, and we've done some work, but we've not published this, is that they can't migrate. So what people have shown is that there's also an alteration of chemokines. So not only are there intrinsic defects with the cells, but the environment which they sit in is also being altered. So we wanted to look at this further, and I just wanted to show you this really interesting study where we've been immunizing mice and looking at the microenvironment. And I just want to show you a couple of pictures. This one in particular. And so we're using an antigen that's been commonly used in immunology from the 60s and 70s, TNP-5. So everyone's been using this as a real nice model to study immunization. I just wanted to show you just how interesting in terms of an immune response. And what you can see in young mice, these are about 3 months of age, you can see 3 days after immunization, you can already see an increase in this is in IgM. Whereas in old mice, you see the difference. Old mice are over 18 months of age. It takes them 7 days to see an increase, a significant increase. And that's what immunosenescence means. And similarly, when we looked at an IgG response, and what you can see, for example, is that upon secondary immunization, again, young mice will see an increase in their immune response with this antigen, TNP, KLH, but you don't see that increase in secondary immune response in older mice. And this is also then reflected in the microenvironment. So the microenvironment is not effective sufficiently to allow this robust immune response to take place. And in particularly, dendritic cells are completely altered in this process. So let me summarize this part, and I want to end on this idea of longevity and really the importance that companion animals may play. So the environment is altered with age. And one of the important things when we're looking at rejuvenation of the immune system, and this is really interesting, the work that's really been able to show you can enhance thymopoiesis in aging mice, and I think they've also done this in primates, the most effective ones have been the ones that they've impacted on the microenvironment. And I think that's really interesting. So I think more targeting work in that area. The other part I want to say is that it's evolutionary conserved, and what does that mean for the host? And therefore, could there be sort of molecular pathways? And that's something that's really been interesting for me, and that's something I want to discuss, and the idea that the splenic microenvironment. So here's what I want to spend just a couple of minutes before we get our amazing speakers. It's intrigued me. I keep looking at this picture. My students think I must be mad because I just stare at the screen all the time. Could I ask this question? Why is this process evolutionary conserved? And so we published this idea over 10 years ago. And it created a bit of a fuss. But the idea, we said at the very beginning, when there's the thymus numbers decline, and you see this in a number of species, perhaps that's related to some idea of growth. And Graham and I were talking about this the other day. Making T cells is incredibly energy demanding. So maybe for a host, the most important thing, given the fact that, we are surrounded by microbes, so really important that the immune system is important for health. So the important thing, maybe, from an evolutionary point of view, is you just make all your immune system, make all your T, make all your B cells, and then afterwards, it can decline. Something Tom Kirkwood and others talk about the disposable soma theory. So we came up with this idea that it's perhaps growth, but also associated with lifespan. And indeed, when we started to compare dogs, particularly short-lived and long-lived breeds, we noticed there's a different rate of thymic involution. Short-lived dogs, thymic involution is incredibly rapid. Long-lived dogs, the rate of decline is less. And here's something that's really provocative. So a colleague of mine, Richard Aspinall, published a paper a few years ago and said he's been doing some work again in humans. So this is really provocative. And he said one of the reasons why perhaps women live longer than men is the fact that the rate of thymic involution in women is actually less than men. And he's been publishing this and talking about this. And that's really interesting. So is thymic involution rated with longevity? And this is more data to show, again, the rate of decline significantly differs between long-lived and short-lived. And so to look at this idea about linking with aging and longevity, I came across a paper several years ago about this idea of developmental origins of adult diseases, that nutrition or environmental stress that impacts the in utero has actually an impact on longevity. And this is some work by Susan Ozanne and colleagues at Addenbrooke's in Cambridge, where simply just having an alteration of protein diet, whether either gestation or sort of during the suckling period, has a significant impact on lifespan. So this is just normally control, whereas just having just 8%, you'll see lifespan is significantly reduced. Whereas actually having 8% during the suckling period, lifespan is significantly increased. So I saw this data and I sent her an email to say, can I look at the thymus of these animals? She works on kidney, etc. Initially, she didn't reply because she didn't know who I was. So I just camped outside her office. I think she got really scared. Who's this geezer that I keep, So my advice, don't stalk people, right? It's kind of scary. But eventually, I convinced her. And here's what we find. And this is something that's really interesting. And we've published this. And we want to get some more money for this. And that's the reason why I'm here. So here's something that's really interesting, is the fact we decided to look at mice very early. We looked at mice 3 months of age. And here's the interesting thing. Basically, at 3 months of age, the short-lived animals already have a signature within the thymus to indicate that they're actually going to have thymic involuting at a much greater rate. So if you look, for example, at molecules like PCNA or longevity factor or P16, it's just remarkable that at 3 months of age, they were already having a signature. So whatever is defining aging at those early stages of life is also having an impact on thymic involution. And interestingly, that relates also to the periphery. What you see in long-lived animals is that they still have more naive T cells in long-lived animals compared to controls, or compared to the animals that are short-lived. So what's happening at the early stages of aging, which related to thymic involution, if you alter in thymic involution, you alter all the phenotypic steps subsequently. And this also relates to inflammation. So what you see, particularly in animals that are short-lived, at 18 months of age, they've got a much higher level of these pro-inflammatory cytokines. So this idea of the impact of early life. So this is what we think is going on. This is my final slide. So here's what we're thinking is going on. So during development, if there's an alteration of nutrition in early life, and there's loads of studies that have looked at, I'm sure you guys are all familiar with this, more so than I am, it is the fact that if in these recuperated mice, which are short-lived, basically it's impacting on the thymus. So there's an accelerated thymic involution and there's an accelerated immunosenescence. Whereas diet that's predisposing animals to have an extended lifespan, for whatever reason, and we still can't work this out yet, thymic involution still occurs, but it's at a much slower rate, and there's a delay in immunosenescence. But what this all means is that the immune system is programmable in early life. And I think that's something really worth exploring. And if there's a way of altering sort of the trajectory of immune aging in early life, that I think is going to be fascinating. And the use of companion animals, which I think can shed important light. So these are all the people that have been responsible for this work. And I'm really grateful again for the various funding agencies. So what I've spoken about thymic involution and its impact on peripheral immunity, and Dr. Dugal will be speaking about that. I spoke about immune aging, and I spoke about the idea of inflammation and the phenotypical changes. And Professor Powlett's going to speak about that. And then we're going to also hear from Dr. Charles, who I think has done some fantastic work about diet and immunity and immune senescence. So that's enough from me. And what I'm going to do, I'm just going to allow Dr. Powlett to take the stage. Professor Powlett, thank you very much.